From the procedure of the experiment, a talent show leaves lower epidermis was peeled off, then placed on slides with water and salt solution. Then we observe under a microscope to compare stomata to showing that stomata are open in water and closed in salt solution due to water loss from gut cells. For the results, we observe stomata under two conditions. The first one is distilled water and the second one is 10% salt solution. Under distilled water, the stomata appeared open at both 10 times and 100 times magnification, showing that gut cells were turgid and allowed water loss. In contrast, under the 10% salt solution, the stomata were mostly closed as the gut cells lost water and become flaccid. This shows that high salt concentration causes stomatal closure to reduce water loss in plants.
In this experiment, the process of water transport in plants was studied using celery stalk. The procedure was carried out as follows. A razor blade was used to cut about 1 cm from the bottom of each celery stalk while it was submerged in water. The cut ends were then placed into 3 flasks containing fusion dye. Flask A held a stalk without leaf, while flask B and C contained stalk with leaf. Flask A and B were kept under normal laboratory condition, whereas flask C was placed in front of a running fan with a lamp positioned about 15 cm from the top to warm the leaf. All stalks were left in their respective condition for 30 minutes. Afterward, they were removed, rinsed under running water, and placed on tissue paper. Using a razor blade and ruler, 1 cm segment were cut from the bottom of each stalk to observe the presence of purple dye in the xylem. Cutting continued until the dye faded, and the distance travelled by the dye was recorded for each stalk. Finally, the results were analysed and explained. Table shows the result for part B, which is water transport in plants. For flask A, placed under normal condition without leaf, the dye travelled a distance of 2 cm. For flask B, placed under normal condition with leaf, the dye travelled 4 cm. Meanwhile, in flask C, which was placed under a lamp and in front of a running fan with leaf, the dye travelled the furthest 6 cm. This shows that the presence of leaf as well as factors like light and wind increase the rate of water transport in plants.
The answer for question 1 is the opening and closing of stomata are controlled by the water pressure in gut cells. When the gut cells are full of water, the stomata will open, but when they lose water, the stomata will close. This process is affected by light, carbon dioxide, humidity, and the hormone abscisic acid. The answer for question 2, environmental factors like light, temperature, wind, and humidity affect water transport by changing the rate of transpiration. Light and heat increase water loss, wind speed it up, and high humidity slow it down. For question number 3, the term circulation does not apply to plants because water moves only one way, from roots to leaf through xylem, and does not return. This one-way flow is caused by root pressure, capillary action, and transpiration pool.
In this experiment, celery stalk in class B and class C were exposed to different environmental conditions to observe how transpiration affects water transport. Class C, placed under a lamp and a fan, showed faster dye movement due to increased transpiration. The experiment could be improved by testing each factor separately using multiple samples and keeping other conditions constant for accuracy. When a plant faces water stress, it closes its stomata to reduce water loss. The hormone abscisic acid, or ABA, triggers the response by making gut cells lose to grow pressure. Some plants also adapt by rolling their leaves, having a thicker waxy cuticle or smaller leaf to minimize evaporation. This experiment shows that stomatal opening and closing control gas exchange and transpiration. In distilled water, gut cells swell, opening stomata for photosynthesis, while in salt solution, they shrink, closing stomata to reduce water loss. This demonstrates that water movement in gut cell regulates stomatal function and the plant water balance. In the second experiment, water moves up celery stalk through the xylem due to transpiration pull. Flask A without leaves shows little movement, while flask B with leaves under normal conditions shows upward water flow. Flask C, exposed to heat and airflow, shows the fastest movement, demonstrating that leaves and environment conditions affect water transport.
This experimental approach helps us better understand the structure and function of stomata by allowing us to observe them directly under the microscope. By comparing the epidermal pils treated with distilled water and salt solution, we could clearly see how stomata open and close in response to different conditions. This hands-on experience reinforced our understanding of how gut cells regulate water loss and gas exchange, linking the microscopic structure of stomata to the important role in plant physiology and water regulation.
For part B, through this experiment, we gain a better understanding of how water moves through the xylem in plants. By placing celery stem in chlorinated water under different conditions, we could observe how water travels up the stem. This helped us connect the theory of transpiration and capillary action with real experimental observation, making the concept easier to understand.
The challenge we faced in part A, at first it was difficult to peel the epidermis and prepare the slide properly. The tissue often tore or wrinkled. We overcame this by working as a team and asking the lab assistant for guidance on handling the leaf peel carefully. With cooperation, we successfully observed the stomata.
For part B, one challenge our group faced was being ensure how far the color water has moved up the celery stem, since it was not very clear. We asked the lab assistant for the guidance, and he advised us to continue cutting 1 cm segment as long as even a small amount of color was visible. This helped us obtain more accurate observation. From this experiment, we learned the importance of careful observation, teamwork, and communication when conducting experiments. It also showed us that seeking guidance is an important part of problem solving in science.